Mira Fayez, and I'm a member of Ratio Christi. And as you guys know, Ratio Christi is Latin for the reason of Christ. We believe that Christians fail to respond to pressing questions today about philosophy, science, and other fields of academia, even though we're perfectly capable of doing so. Ratio Christi equips people to stand for what we believe in, and it addresses these questions. If you guys want to know more information, go to Facebook and like our page at Ratio Christi UC Irvine. Today is the final week of our ID discussion, and we will be meeting from now on here at 6 still, but with a different series of ones. Next week, we're going over tactics to defend the faith. And on November 6th, we will have a focus group, and participants will be paid $20 to participate in the focus group if you guys are interested, and bring friends. Today we have Ryan Huxley, who will finish our Intelligent Design series, and he will be guiding us on an exploration on how the biological design model applies to intelligence design and about intelligence design research. He is the president of the Intelligent Design and Evolution Awareness Center. He has a bachelor's degree and a master's of science in engineering from UCSD. He's a licensed structural and professional engineer. And has lectured in multiple lecture halls about engineering as well as intelligent design. He's been involved in intelligent design since 2002 and has led many lectures and courses, conferences, and other panels concerning these issues. Please join me in welcoming Ryan Huxley.
Thank you very much for that introduction. Appreciate it. Oh, yeah, these the links. Thank you. So as she noted, we're going to be talking about intelligent design and biological design. So that's going to be the primary focus for tonight's talk. This is going to be the last of a 4-part series for those of you who are interested. We started off about 4 weeks ago with Casey Luskin talking broadly about intelligent design and the scientific method. Then there was Tyler Geffany with Lester Media, who spoke about cosmological design and fine-tuning. And then last week was Stuart Orr with the Idea Club in Orange County, and he spoke about the origin of life and intelligent design. So now we're going to get into biological design. So I'm not going to bore you with this since you've already got the introduction about who I am, but to talk a little bit more about the Idea Center and the group that I'm with. Some of you are probably already familiar with us since I've spoken previously here. So one of the main reasons the Idea Center exists is to promote intelligent design as a scientific theory, because that's something that is becoming more and more difficult to do these days, especially when it comes to academic freedom. And depending upon how much time we have, we can perhaps cover that later on. So one of the other things that we do, or in the process of doing that, we educate people about intelligent design as well as scientific and philosophical challenges to naturalistic explanations for life. And we try to help students form idea clubs so that they can have discussions about origins topics, hopefully in an atmosphere where people will not feel like they're going to be jumping down their throat about it. And that's been a little bit more difficult as the years have progressed because of the academic intolerance and the lack of academic freedom. So we help students form idea clubs to do a lot of these things that you see here. So a little bit more about idea clubs. If you're wondering what they are basically extracurricular clubs that provide a forum for students, faculty, and community members to explore and discuss and debate the scientific evidence regarding intelligent design and neo-Darwinism. So in a sense, it's the same kind of thing that's happening right now. This is often what is involved with idea clubs, but they often will just have group discussions about these topics. All right, so let's go on ahead and talk about, give you an outline for what we're going to be going over tonight. So the first thing we need to do is define what neo-Darwinism is. Then we're going to investigate neo-Darwinian mechanisms driving biological change. Next, we're going to investigate whale evolution based upon the previous two items. And then last, we're going to look at, based upon what we observe, do we have evidence for intelligent design or neo-Darwinism? So let's go on ahead and get started. So what is neo-Darwinism? Well, you likely have a pretty good idea here, but the quick answer is it's basically just biological evolution. But it's important, before we even think about that, to really define these terms like this, because in the process of defining the terms, we're making clear what topics we're really discussing, because evolution can have a lot of different meanings. So it'll help us to make sure that we're talking on the same page here, rather than, wait, you're saying this and I'm saying this. So it's really important to go on ahead and be sure to clarify exactly what is it. So as I mentioned, it's biological evolution, or more technical definition is that it's the modern coupling of Charles Darwin's theory of natural selection with Gregor Mendel's inheritance ideas in light of genetics. Now, it turns out that this is also known as the synthetic theory, and it actually came around quite a long time after Darwin and Mendel did. Back in the 1930s and 40s, this is about the time that the modern synthetic theory, as neo-Darwinism is also known as, came about. And it was actually, the term synthetic theory was actually coined by a fellow named Julian Huxley. I don't think there's any relation. So he coined the term with a 1942 book that he wrote. And he joins the disparate fields that I've already mentioned, plus quite a few others. And those are things like paleontology, systematics, this is how you categorize different animals, and morphology as far as the study of gross features, physical features of animals, ecology, and so on. Because those fields were beginning to have more data that they were gathering from the observable world at the time, and so they're needing to try and bring all of these different fields together. And that's what the synthetic theory did. That's why it was a synthesis of all of these different topics. So the key topics of interest, really, that we're going to be talking about are Darwinian natural selection with Mendelian genetics to arrive at mutation selection mechanism. That's what neo-Darwinism is really often dealing with in our discussion tonight, and often the way that people refer to it when talking about the debate between intelligent design and neo-Darwinism. So Mendelian genetics provides Darwinism with the naturalistic method of inherited traits. Because at the time that Darwin wrote his Origin of Species, he did not have a mechanism for how things got inherited. They did not have anything like that. They didn't have an understanding of what that was. So he came up with the idea of natural selection, but it wasn't until later that Mendel, through his understanding of inheritance through studying peas, that was really what the driving mechanism was for inheritance. Now, they were doing things at about the same time. Mendel was a little bit later than Darwin, but people didn't actually recognize these as complementary ideas until many years later. So there's other names for the modern synthetic theory, which are listed here. So the new synthesis, the evolutionary synthesis, millennium synthesis, and the neo-Darwinian synthesis. So what exactly are neo-Darwinism's main claims? Well, all organisms are related through common ancestors. That's one of the chief claims of neo-Darwinism. That's also how people often think of evolution. That's one of the definitions often given. And another way to think of this is that all organisms and their genes arose through blind and unguided processes, which are random mutation, undirected natural selection. By the way, natural selection is not random. And then random genetic drift, which has no impact on survival. So these are some of the things that are associated with explanations to explain the diversity of life that we see today that has occurred throughout history. So this is the basic idea. all organisms arose and persist because of the blind, unguided, and random processes of nature. That's the nutshell summary for what neo-Darwinism is. So I like this because it provides, I'm a visual person, and so rather than having lots of words, as they say, a picture is worth 1,000 words, so this provides a great description of essentially what I just attempted to describe with a few words. And it also provides a great little summary of some of the things that have been covered over the last few weeks. So starting in the upper left corner there, you see initially the Big Bang essentially, which then gave rise to all of the different stars and galaxies and planets and so on, and eventually you have our star, the sun, and then with about the same time the Earth, and then you have the Earth in its initial states as you go down on the left-hand side with a lot of volcanic activity, very violent time in the Earth's history, and then you have a lot of this chemical soup, prebiotic soup. You keep on going down the left, and somehow they start to come together. You start to go from the left to the right, start to form these interesting molecules, and eventually somehow give rise to the first form of life, keeping on going from left to right. That life becomes more complex, start to get very simple unicellular life down here in the middle part, and you're starting to get some of the cyanobacteria above that sort of white line or curvy line, and life continues to become more complex as you start continuing to go up the path here, start to get more complex features, vertebrates and fish, and then you have amphibians, and you keep on going up the path here, some reptiles and dinosaurs, and there's early humans, and you keep on going up, and then you have the telescope, modern man, who's then looking up in the sky and looking back in time to see this all occur. So that's sort of a quick nutshell summary there. All right, so now that we've defined what neo-Darwinism is, let's look at the specific mechanisms that are often given credit for driving biological changes that have occurred over time. So before we do that, we always need to have, again, a little bit more background to explain the context for what it is that we're going to be discussing. So let's talk a little bit about biological organization, because that'll help provide a better understanding of why we're going to talk about the particular features of naturalistic change. So when it comes to biological organisms and biology, there's a way that biologists have organized life. It recognizes the different levels of life, organisms from small to large features. The cell is really the basic unit of life. That's something that can survive essentially on its own. And if you were to try and define life, a good explanation or definition of it for life is that it has self-sustaining and signaling processes that non-living things do not have. And they can process energy, grow, respond to stimuli, and maintain internal equilibrium and replicate. Of course, that replicate part is one of the key features through which inheritance, genetic changes, and so on provide us with supposedly new features. So when it comes to cell organization, there's two different types. There's prokaryotes, which generally is bacteria and things like that, and there's eukaryotes, plants and animals. That's essentially what eukaryotes are. The eukaryotes have organelles in them that are definable, things like the cell nucleus, the Golgi apparatus, vacuoles, things along those lines, mitochondria, endoplasmic reticulum, things like that. So these are features that are smaller than the cell. These are inside of the cell. Prokaryotes don't have very well-defined internal features. Now, both of these do have DNA. Bacteria has DNA, and so do plant and animal cells. Both of them make use of proteins. Proteins can form structures, which are really oftentimes nanomachines. We'll see that a little bit later. Probably the most famous currently in the ID movement is the flagellum, the bacterial flagellum. It's a rotary motor. We'll talk more about that later on. It's sort of the poster child right now. Then there's ATP synthase, which is also another nanomachine, there's a kinesin motor, and so on. They also can form enzymes, which essentially provide the ability for a cell to metabolize different things, so they interact with other biomolecules. There's also a cell membrane, which is mainly in eukaryotes, whereas it's a cell wall for plants in particular. And then there's organelles, which I already talked about in the eukaryotes, and then there's a cell nucleus, again in the eukaryotes, and that's where the DNA is generally stored. So for features that are larger than a cell, generally when you start to group cells together, say at least with animal cells, when you start to group those into larger colonies, if you will, they form tissues. That's the terminology that we use when you start getting groups of cells together. And then when you start to get groups of tissues together, you get what are referred to as organs. And then when you get organs together, then you get systems. So this is looking at larger and larger organizational levels for biology here. All right, so the central dogma. How many have heard of this idea? All right, so the central dogma is a description of what happens with DNA and how it makes proteins. So the idea is that DNA makes RNA, makes protein. That's a little bit of a simplification there, but that's the way that it's often, I guess, memorized, so to speak. And what this is really saying is that the genetic code dictates the amino acid sequences that make up proteins. So DNA has these codons, these segments of DNA that dictate a particular amino acid, which is then strung along in a long chain, and when this chain goes and becomes folded into a certain shape, it becomes what's referred to as a protein. So proteins are used in metabolic pathways and subcellular components and biomolecular processing, such as enzymes, which I mentioned previously. And later on in this presentation, we'll see some videos because for those of you who are not biology majors, this is going to be a lot of new jargon that's difficult to follow. But just understand the basic ideas that we're talking about subcellular components right now. So when it comes to genetic mutations in proteins and their impact, or genetic mutations and their impact on proteins, the basic idea is that depending upon the DNA mutation will cause some type of change in the protein because it's modifying the amino acid sequence in some way. So depending upon where that location is, where the mutation is within the DNA, it may or may not have a substantial effect on the protein. It could affect its ability to fold properly, or it could affect its ability to properly function. It sort of depends upon where that mutation takes place for the particular amino acid sequence or protein that is being impacted by it. So this is really where the rubber meets the road when it comes to neo-Darwinism, all right? So genetic mutations are what bring about modifications to a sequence of amino acids, which then bring about modifications to the proteins. This is where you're seeing change occur at the molecular level. All right, so just to, again, instead of having lots of words, just to give an idea of what we're talking about here, this is a quick little animation of subcellular activities in other larger biological organisms. So let's see how the sound works here. so you get a little bit of an idea there of what we're talking about. So that hopefully provides the context for what we're going to be talking about here. So, what are the key claims when it comes to Neo-Darwinism? Well, it is that genetic mutations bring about amino acid sequence modifications that bring about new proteins. So there's going to be some type of new function or new folding that occurs. Now, if that's the case, then what this means is that there are 5 questions that really need to be considered here. And this is really as a result of the logic demanded by Neo-Darwinism. First one, are the claimed changes possible through mainly genetic mutations? That's the first question. Second question, is there a stepwise adaptive pathway to mutate from protein A to protein B in which there's a selective advantage that is being provided at each small step of the pathway? Now, if not, if that last question, if the answer is no, then are multiple specific mutations required to gain or improve function? And if they are such multi-mutation events likely to occur given the available probabilistic resources? Is it even feasible with the kinds of mutations that are required? And then last, can mutation benefits be tied to the paleo environment? In other words, can we tie these new features and these supposed benefits to what was occurring in the ancient environment? So why is it important to ask these questions? Why do we need to consider these? Well, answering these questions are fundamental to the way that Neo-Darwinism works. Right? This is fundamental to their claims. But these are often lacking, as far as answers to these questions are often lacking in the supposed evolutionary explanations. And there's a biologist here, I'll just read the quote, who acknowledges this, evolutionary biologist Austin Hughes. A major hindrance to progress has been confusion regarding the role of positive Darwinian selection, that is, natural selection favoring adaptive mutations. In particular, problems have arisen from the widespread use of certain poorly conceived statistical methods to test for positive selection. Thousands of papers are published every year claiming evidence of adaptive evolution on the basis of computational analysis alone, with no evidence whatsoever regarding the phenotypic effects of allegedly adaptive mutations. Phenotypic is, that just refers to not only the physiological modifications, but behavioral modifications that may result from a particular mutation event. So it's sort of a catch-all that captures all aspects of a particular genetic change as far as jargon here, as far as phenotype. So evolutionary biologist Austin Hughes further indicates that it's important that biologists should avoid making statistically based claims of evidence for positive selection that is divorced from any biological mechanism. Oftentimes you'll see in the literature that, oh, this seems to be something that likely worked better, so therefore evolution explains it. That's not really sufficient. We have to go through and answer those questions that were outlined previously. He further laments that even in sophisticated studies, there was no direct evidence that natural selection was actually involved in fixing adaptive changes. That's a pretty severe charge. And that this person is, as far as I'm aware, not an intelligent design proponent. He's not friendly to ID. But he's recognizing the lack of rigor when it comes to providing full descriptions that are plausible to explain what is observed. So, in a sense, what we're asking is, show me the numbers. That homology, which is essentially a fancy word for similarity between different organisms, they'll use homology of sequences or structure for a given protein. And then they will start to invoke these explanations, things like gene duplication, divergence, exon shuffling, DNA rearrangement, which includes things like insertions, deletions, retrotranspositions, etc., lateral horizontal gene transfer, That's where supposedly one organism is swapping its genes with another one. And they do see something like this at the very simple organismal level as far as bacteria are concerned. But then there's also de novo creation, which is a novel sequence, and there's also these orphans. Orphans refers to open reading frame sections of DNA. That's what the ORF is for. But essentially, they're starting to use these other supposed mechanisms to describe these more complex features to try and explain what they're seeing. So the basic idea is that they're postulating a method for DNA mutations and modifications that are usually involving larger scale changes to the DNA sequence. That's what these ones that are listed up here are really doing. They're more than just point mutations. This is more than just one little step along the path here. So these mutations then are said to be affecting either the amino acid produced or the sequential location, and then of course this affects the proteins. So we're going to consider now this idea of de novo creation, novel sequence, and orphans, because remember that those are unique. That means that they do not find similarity with anything else. So it's a problem then from a common ancestry perspective. So, how did the change occur? Well, while these proposed mechanisms that I noted there may be interesting, they're really not very descriptive other than saying, well, there's some fortuitous events that occurred and it just happened and that's how we got the change. So there's really not much beyond what I just noted there, these fortuitous events that are being given to describe exactly how new DNA sequences arose. So what is lacking when these kinds of descriptions are being given? Well, starting to get into some of the answering the questions here. They're not providing the actual stepwise descriptions to clarify the pathway from gene A to B, or protein A to protein B, including things like the intermediate functional characteristics, how these new features were properly integrated into existing biochemical systems or metabolic pathways, whatever it is that they're talking about, whatever this protein's role is, and then providing a statistical justification demonstrating the feasibility with the known probabilistic resources available as far as how many organisms do you have and what time frames do you have based upon population genetics. That accounts for the number of organisms, the mutation rate, the generational period, and so on. And then based upon that information, is it plausible? Is it reasonable? So when it comes to de novo and creation and orphan segments of the DNA, this is a little bit problematic because de novo genes are essentially genes that seem to arise out of previously non-coding regions. How did that come to be? Well, they just say, oh, it doesn't happen. Orphans, these are unique stretches which are unlike any other known protein coding sequence. And it turns out that when it comes to orphans, at this point in time, they make up 10 to 20% of the currently known genomes. How that number may change over time as we continue to decode the genomes of different organisms, time will tell. But one thing that they have noticed is that these orphan sections of the DNA are frequently species or group specific. They seem to be conserved within particular groups or subgroups or species. Now this is going to be difficult to explain then if these are unique aspects, because then you cannot cite common ancestry for how these arose, because they're unique. They're not like anything else. So remember the first question was, is the change, or are all aspects of biology dictated by genes, or can genes dictate all changes? Turns out that are there any aspects of biology or biological change or development that are not solely dictated by the DNA and genes? Yes. The catch-all term is epigenetics. That term basically means anything outside of the genome. So when it comes to early embryological development, it turns out that genes are not the sole descriptor or director for what things are going on. It turns out that there's a complex interaction between the three-dimensional structure of the cell, biochemistry, and genes. There's this complex interaction between them all. They're still trying to figure out exactly how it works. So as far as cell differentiation and specification, again, this is within embryological development. As far as the body plan layout, these things are not defined solely by the genes. There's some other information systems involved here that are describing the development of the organism. So what is the implication of this? What it means is that if you're going to look to genetic changes to describe modifications at this level when these kinds of things are occurring, it's not going to be very successful, because the genetic change is insufficient to achieve the desired outcome, because there's far more involved with the development of the organism than just genes. So what that means is you can mutate something as many times as you want. You're not going to be able to end up affecting the body plan layout by that, except perhaps cause lethality, cause death, basically. So if you'd like to get some more resources on epigenetics, there's a great Illustra Media film, Darwin's Dilemma, talks a little bit about this, where it talks about the Cambrian explosion and the animal phyla that arise during that particular period that came during the Cambrian period. There's also a discussion on a study guide that goes along with that in Chapter 5, is where it talks a bit about these epigenetic factors. And Stephen Meyer's Darwin's Doubt, which just came out this past summer. Chapter 14 deals with this. All right, so dealing with the second question. Remember, this is the question about, is there a stepwise pathway to describe to go from protein A to protein B? So this is actually an image from that Darwin's Dilemma film, and it's essentially showing the idea of Dawkins' analogy with the climbing mountain probables. So you see these steps that are climbing up the mountain there, and there's a cliff on the edge. But so of course no one would say that to arrive at the top you must have somehow scaled the cliff. From a genetic standpoint, they would say that there's just these slight modifications and you just slowly walk up the hill. That's the basic idea for stepwise path to get from one protein to the next. But could there be a problem if there's no stepwise path available? Yes, clearly there would be, because it's going to rob the creative efficacy or power of natural selection. Because natural selection is often, again, given credit for providing the engine for change through these minor modifications. So the second and third question, rephrased essentially, this is the stepwise pathway, and if there's not a stepwise pathway, then you need to have multiple mutation features. Rephrasing those two together, how common are amino acid sequences leading to functional proteins? This was probably looking a little familiar for those who saw Casey's presentation. And so I'm using some of the same information that he talked about, but applying it with respect to the key questions to deal with when looking at neo-Darwinism and the mechanisms often discussed as being, or given credit for the changes that are seen. So, how common are these amino acid sequences? Well, as you can see here on the x-axis is the sequence space. These are all of the possible amino acid sequences you could have. And then on the vertical axis is enzyme activity. And based upon some of the mutational sensitivity studies, as well as theoretical and numerical simulation or studies that were done, Doug Axe found that it turns out that there's these like little islands of functioning. So you have something doesn't function, all of a sudden you make a minor modification, boom, suddenly it's working. It's along the lines of an all or nothing situation. So you can't just get any modification to a protein and expect that you're going to have things functioning properly. That's why this is important. That's why the studies that Doug Axe did are important, is that he showed that properly functioning or folding proteins are rare. Okay, they're not just, all I have to do is mutate a little bit here and there, and you get another one, and mutate it, keep on going. You're just having a continual functioning protein. These studies that he did show that that's not the case. So amino acids must be finely tuned. And for those of you who saw the talk that Tyler Defany gave on fine-tuning with the universe, we're seeing the same kind of fine-tuning when it comes to protein folds. So for functional protein folds, they're very finely tuned, and they are essentially multi-mutational features then, because you can only get from one island to the next island. You're going to have to go through a lot of mutations to get there. So it turns out that based upon another paper, not the one that's noted here, but another one that Doug Axe did, which will be in the next slide, he found that the odds of getting a functional protein fold could be as rare as 1 over 10 to the 151st. That's very hard to imagine, okay? That would be for a typical sized protein with about 300 amino acids to it. And that's noted in this paper. So, in this paper, what Doug was doing was he wanted to determine what was the power, what was the potency of the neo-Darwinian mechanisms, and also consider the sampling problem as far as all of the available options that are out there. You need to make sure you sample the right one to get a functional protein. So, based upon the work that he did in that paper, he came up with this maximum feasible search by neo-Darwinism, the mechanisms associated with that for an ancient bacterial population is the maximum chance that you can actually get something by chance is 1 in 5 times 10 to the 23rd. That's if you were to have a decent sized bacterial population that had so many generational periods during the course of a year multiplied by the age of the Earth, about 5 billion. So you can go to that particular paper to see where that number comes from. So, in a sense, what that means is that the chances of getting a common length 300 amino acid protein range from about 1 times 10 to the 151 down to, in the paper he notes, about 1 in 10 to the 104th, depending upon what kind of situation you're looking at. So what does this mean then? Well, it implies that the ability to arrive at a functional protein then, you just take all the possibilities divided by the number of functional sequences, and you can see you're going to end up with something far less than 1. That means you're not likely to get there through point mutations. Okay, so maybe we're not being generous enough here, all right, because we were using 300 spots. All right, let's be more generous then. Let's be more conservative in our estimates here. How about all life forms that ever existed and a shorter protein, say only about 150 amino acids long? Does that then get us there? Okay, well, how many life forms have there been? Well, there's been about 10 to the 40th total life forms that can try different amino acid sequences. Let's just say that they're all mutating, even though that's not really the case. It's usually about, at least on one of the papers it noted about 1 in 300 where that you'll see some modifications. And based upon using this reduced number of 150, roughly, amino acids, the chance of getting a functional protein of length about 150 is 1 in 10 to the 77th. We're still far less than 1. So what does this mean? This means that there's basically not enough probabilistic resources to get a functional protein by chance alone if you are going to try and describe it through point mutations. These are the small sequential steps. Now, you'd think, okay, is this only intelligent design proponents who are recognizing this? No. Others are as well. And this is noted in Campbell's Biology, a popular college-level biology textbook. Even a slight change in primary structure can affect a protein's conformation, that means its fold basically, and ability to function. only a small fraction of the possible combinations of amino acids will fold spontaneously into a stable structure. If you make a protein with a random sequence of amino acids, chances are that it will only form a gooey tangle when placed in water. So they're recognizing that there's some problems here. Again, basic idea, functional protein rarity implies fine-tuning of useful amino acid sequences in DNA coding. All right, let's talk again about the third question as far as multimutations. We can rephrase that. How quantitatively different are similar proteins? Well, Doug Axe and Ann Gager of the Biologic Institute, Pro-Intelligent Design research group, has published a paper a couple years ago, in which they looked at what are the chances of being able to change, well, they did some experimental work where they tried to change one protein into another protein. They were structurally very similar, and those particular proteins was the KBL and BioF, and what they're doing is they're testing some of the mechanisms that were quoted previously, things like gene duplication and co-option, to try and describe how you can get the protein evolution. So this is from that paper. So we have these two proteins here. So this is the structure for the KBL protein, and this is the structure for the BioF. And you can see they look very similar. They're not identical, but they are very similar. So, very closely related enzymes. So how many mutations are required to go from KBL to BioF? That's the question. Well, it turns out that based upon their studies, they found that you needed to have at least 7 mutations, and it's very likely that you probably need to have even more than that to arrive at a functional, if you were to change KBL to BioF. So this is an example of a multi-mutation feature. Now they have a number. What does that number really mean? Well, in order to make sense of that number, what you then have to do is you have to start looking at what are the probabilistic resources available. How many organisms do you have? What's the mutation rate? What's the time that you have? What's the generational period? And that's what population genetics provides. It basically helps us answer the question, are the identified number of changes feasible? Well, we have some more work that's been done by Michael Behe and David Snoke, in which they looked at whether or not unguided evolutionary processes can add novel proteins to functionally interface with existing proteins. Again, they're testing gene duplication claims. So they're trying to find out, again, are the claims made about these Darwinian mechanisms, are they realistic? Well, in answer to that question, what they found was that additional mechanisms must be necessary because based upon the point mutation model that they looked at, there's just basically not enough time. You're going to need to look to other neo-Darwinian processes beyond the point mutations in order to generate many of the protein-protein interactions within the time frame and anticipated historical populations. So what are they talking about? Well, recall those vague descriptions that are larger scale as far as the kinds of changes that are occurring. These are not step mutations. These are insertions, deletions, things along those lines. This is not mere tinkering, okay? This is where you're getting wholesale modifications in your genetic code sequences. And if we think back, so this is going to be what's going to be necessary because of those rare point locations or rare island locations for a functional protein. You're going to have something that can jump across that valley of non-functional proteins. So we're talking about some significant changes. So this graph basically shows what Behe and Snoke found in that paper I mentioned. What they found was that based upon a given population size, so this is the number of mutations that are required on the x-axis, and on the y-axis is the time to fixation. And what they found that was based upon the time frame that they were investigating, which is, that's what is the horizontal line coming across there, that you would only be able to get maybe 2 mutations. And that's within the entire history of the supposed common ancestor or supposed development of this particular organism that they're looking at. That's a pretty low number. So if there's more than 2 simultaneous mutations that are necessary to produce a functional protein-protein interaction, it is unlikely to evolve in multicellular organisms on any reasonable time scale. So they were looking at multicellular organisms here, more than just a simple bacteria. So when you're thinking about mutations, you really need to consider, okay, what are there different kinds of mutations? Well, yes, there are. You really need to look at all the different mutations you could have. There are those that are considered adaptive, that are those that are, as you would think, those are the ones that are helpful to survive, provide some type of net benefit. And it may not necessarily be that it's necessarily becoming more complex. We'll talk about that a little bit later. There's maladaptive, which means that the mutation is actually harmful to survival. Then there's neutral. So it's not providing a benefit nor being harmful. So there's several different options for how this modification then can become fixed in a given population, how this genetic change can achieve fixation. And that's through something referred to as molecular saltation, and that's essentially that you're going to sort of get there in one fell swoop. That's very unlikely. Then there's something called sequential fixation. That's where the entire population goes through this modification, but it's rather unlikely for, say, neutral or maladaptive, because maladaptive, they're most likely going to die off. So really what needs to be, what's probably the most likely one to be consistent with what, say, the Darwinian model would be comfortable with, or evolutionary biology would be comfortable with, is this idea of stochastic tunneling. And that's another jargon term. It's very similar to punctuated equilibrium. Essentially what you're doing is you're taking small subpopulations within a larger population and then allowing those ones to change. It's conceptually similar to punctuated equilibrium. So what is going to be the most favorable then if we're going to try and give neo-Darwinian mechanisms the best chance? Well, stochastic tunneling is the one to pick then. And what he found was that the maximum number of mutations that you could have within the time frame that he was looking at was either 2 if they were maladaptive or 6 if they were neutral. All right? So he went through some heavy math in order to arrive at that, but the nice thing is that he has a nice graph which pretty much summarizes it. And basically, if a multi-mutation feature requires more than 6 neutral, that means that it has no impact on survival. If it's going to require more than 6 or more than 2 maladaptive harmful ones, mutations before it provides any type of benefit, then you're not going to have that arise within the history of life. Kind of a problem. So recall that we were looking before at the KBL to BioF protein. That took at least 7 mutations. So if there's more than 6 are required to produce a new protein function, this is not a new protein fold, just a new function, then it's unlikely that it's going to evolve in bacterial populations on any reasonable time scale. Now, when they were doing the study of KBL to BioF, they were not looking at something that is thought to have been that KBL was a common ancestor, so to speak, for BioF. What they were really trying to do is they were just trying to find out, okay, if you have 2 similarly structured proteins, can you get them to change? Because the overarching idea from a neo-Darwinian perspective is that if you have similar structures, as far as protein folds, the similar features, then you should be able to get from one protein to the next. It should be not that difficult. That's really what they were looking at. So this can be described pretty simply as far as how you go about doing the math for this, all right? So it's essentially the same idea as getting the right combination on a lock, say, by chance. So if you have a padlock with 1-digit combination, what are the chances that you're going to get it right? Yes, 1 in 10, right? All right, how about 2? Yes, 1 in 100. Very good. This is the pop quiz, by the way, for those of you. All right, so all you're doing is you're just multiplying, right? You're multiplying each of these, right? So if you have 4-digit, well, the odds of getting the right one are 1 in 10,000. So how much time do you have to actually get all of these in the right sequence? That's another question that's really important. But this is essentially the basic idea of population genetics, all right? Because you have so many different amino acids. There's about 20 different amino acids. So there's 20 different digits, if you will, that you can choose from. And then as far as which digit that you're looking at, that's the amino acid, that's like the sequence of amino acids. Okay, so if the odds of opening by chance decrease exponentially, right? Exponentially because you saw they were multiplying by each other there. So if the odds of opening by chance decrease exponentially as the number of digits in combination increases, you're going to very quickly run into problems. This is referred to as the combinatorial explosion problem, or the combinatorial problem, or combinatorial inflation problem. So when we have multi-mutation features, that's what we are beginning to run into before you get any benefit. So as more mutations are required, the odds of the feature appearing actually decrease exponentially. This means as far as decreasing by chance, as far as appearing by chance. So this is going to quickly exhaust probabilistic resources. All right, so recall that this is from Doug Axe's work, that if there's at least 7 mutations that are required to change KBL to BioF, it's probably going to be much more. What this means is that this evolutionary transition would exceed the probabilistic resources. What does that mean? That's the number of organisms and the number of generations that are available over life's history on Earth. So it's going to be very hard for evolution to build new features. That's the implication of what this study found. So if they found that to go from KBL to BioF took more than 10 to the 15th billions of years, and yet life on Earth, we're told that we went in less than 4 billion years from bacteria to killer whales, is there a problem? All right, let's consider more about this idea of adaptive mutations. What exactly are adaptive mutations? How beneficial are they? When an adaptive mutation occurs, what is it that what's really going on as a result of that mutation? Well, Michael Behe basically did a literature review, and he looked at about 4 decades of research on this, in this particular arena. And he reviewed laboratory evolution experiments of microbes going back, as I mentioned, 4 decades to determine what seems to be the most common results of these adaptive, remember the adaptive being beneficial, mutations. Well, as a result of that review, he came up with what he referred to as his first rule of adaptive evolution, which can be summarized as this: break or blunt any functional coded element whose loss would yield a net fitness gain. So earlier we were talking before this presentation about Alice in Wonderland, and this is something that Michael Behe has made a joke about. In a sense, what we're talking about here is if this is the way that beneficial mutations are occurring, you could also almost refer to this as Cheshire Cat evolution, right? Over time, eventually all you have left is a smile or something like that. So that's the example Behe gave. I think it's rather appropriate. So it's much easier for Darwinian evolution to break parts than it is to actually build new ones. That's what is found in the literature in which they've done experiments with bacteria and other things like that, and when they find that there's some type of benefit. Now we're going to talk about the last question, the paleoenvironment. How likely is it that we can tie the changes that we see to what the thought to be environment was at the time that these modifications were, say, occurring. So recalling that the fit organisms are the ones that survive, they have some type of benefit or some type of characteristic that makes them more able to survive in the environment that they are in. So how can benefit be defined in the context of the environment? Well, there's several different ways that you'll see this show up. They may say that there's some type of metabolism that is now more efficient as a result of this modification, or there's some new processing ability. Maybe they're able to cope better with the surroundings in some form. But these are rather vague oftentimes. So really, in order to be considered an appropriate answer to this question, there needs to be some specific evidence for the environmental pressures that provide that stress on the organism for selection to then weed out this organism, or to keep it in and weed out the other ones. Simply by having vague references to positive or negative selection pressures don't really have the kind of empirical support to describe the environment that's bringing about this selection pressure. Now, they could be correct, but remember that in order for us to feel that we have any level of certainty in what's being proposed, they really need to have some substantiating evidence to support it. So without providing this and the answers to the previous questions, what we're left with is speculation. Remember that the degree of certainty that we can have for a proposed mechanism is going to be commensurate with the number and rigor of answers to the questions that we've just gone over. All right, now we're going to get into some practical examples with investigating whale evolution. So what are the main claims when it comes to whale evolution? Well, basic history is that the process started about 53 million years ago with some type of wolf-like creature, which is referred to as Pakicetus. This was a fully land mammal, all right? And then whales, Basilosaurus is one of the first ones that's identified. It arose about 38 million years ago, and this was a fully marine mammal, all right? Now, have there been any modifications to this timeline? Yes. Turns out now that they've found some Basilosaurus fossils dating back to 49 million years ago. We've just compressed the time now for all these modifications to occur, to go from a land mammal to a marine mammal. What kinds of changes? Okay, this is what we're talking about here. And we only have about 4 million years to do this. This is a brief list of, I do say brief, this is certainly not all of them, that lists the kinds of changes to be able to go from a land-based lifestyle to an aquatic or marine lifestyle that had to occur over, say, 11 million years or less, probably within 4 million years. So some examples as far as new or modified creatures. They have to have a new kind of spine, new kind of backbone, a ball which includes ball vertebrae. They have to have intra-abdominal testes with a countercurrent heat exchanger. They have to have the ability to intake salt water and actually drink it to get their hydration. They have to have tail flukes and the musculature to control those. The forelimbs must be modified to become flippers. They have to modify the eyes to have underwater vision. They have to have the hind limbs that are reduced. The pelvis and the sacral vertebrae have to be reduced or portions lost. The lung surfactants are modified as well. There has to be a decoupling of the trachea and the esophagus for the proper breathing. There's a modified skull and musculature. The blowhole, the musculature and neurological control for the blowhole. And the modified musculature for reproductive organs has to occur as well. And this is just a brief list that Richard Sternberg, von Sternberg, has come up with, and he has a lot more than this. So, based upon what I went over earlier in the presentation, of the many modifications noted, do we have the probabilistic resources available to achieve the fixity of those changes? Okay, that means that when you have the modifications and mutations that occur, not only do they have to occur in that first organism, but they have to actually be so successful that it ends up spreading throughout and becoming the entire population over time. So, given those required changes, we're going to have either a lot of generations, a high mutation rate, or very large populations. So we're going to have either all of these, or many of these, or some of those, and based upon population genetics and the past reference work by Beatty and Snoke, and using a 5-year gestational period, evolutionary biologist Richard Sternberg found that only 2 mutations would be possible in 43 million years. That's going to cause a problem. So, given that you have multiple coordinated mutations are likely required, 4 million years is not nearly enough time. All right, getting on to the last part of the presentation here. So is what we've seen evidence for intelligent design or neo-Darwinism? Well, it's interesting to think about how materialists resist intelligent design in biology. Here's a quote from Richard Dawkins: "Biology is the study of complicated things that give the appearance of having been designed for a purpose." So it's appearance. It's not real. Biologists must constantly keep in mind that what they see was not designed, but rather evolved. And this is Francis Crick. He's the co-discoverer of the double helix with James Watson, or the double helix of the DNA. So, as you saw from Casey's presentation, intelligent design does make positive predictions. So there are ways that we can test between intelligent design and evolution. It's basically a statistical argument because, and you can see there's sort of a spectrum here that you can see from the left to the right. So given the information content, which is more likely, it's going to be based upon that level of complex specified information, which Casey talked about before. Depending upon how high that level is, then we're going to be ending up more on the right-hand side as opposed to the left-hand side if there's not very much specificity or there's not a very high information-rich content associated with what we're looking at. So when it comes to looking at organisms, there's actually going to be layers of design that we will find. We have encoded information that's in DNA. There's an information processing ability of the cell of that information within the DNA, and the result of that is a product that's typically protein machines. So the key question then is, where does this information come from? How did it get there? That's really the big question when it comes to intelligent design. So with regards to those first 2 layers that we're talking about, that I mentioned, encoded information and information processing, rather than talk about it, I find it's just far easier to use this great video. If I can find it. Oh, that's not what I wanted to do. Let's see. See if I can do this. There we are. So this is looking inside a cell.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, the molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
All right, so that explains in far better ways than what I could try to with that nice little visual image there. So just to talk a little bit more about what you saw there. So with regards to layer 1, this is the encoded information in DNA. So DNA uses the chemical language of the base pairs, A, C, T, and G. All right, so that's adenine, cytosine, thymine, and guanine. And the particular order of those bases is going to determine the particular acid, the amino acid sequence. And thereby it's going to then dictate the shape and function of the protein. So the order is not specified by any physical law or chemical laws. So this is language-based information that we're seeing at the fundamental level of all living things. Okay? We know that language-based information comes from a mind, based upon our common everyday experience of how we know intelligent agents act. They often create information with high information content. So this is what Casey talked about for quite a bit in his presentation. It's the same idea. So thinking more about the information processing that we see within cells. So what the cell is doing is it's converting the information that you see in the DNA into proteins through a process that's called transcription and translation. The translation is that's what occurs when the information in the DNA is translated into messenger RNA, just referred to as mRNA. And then the transcription, this is when you take that information and transform it into a sequence of amino acids. And it's the genetic code where 3 sequence strings of nucleotide bases are going to signify a particular amino acid. And ACG, that's an example of one of the amino acid, that is the coding for threonine and so on. I'm essentially just trying to put into words what you just saw, but I think what you saw is much better than an attempt to try and explain it. Okay, so again, this is the basic idea is the central dogma of biology here. So DNA makes mRNA, and then plus tRNA gives you proteins. So here's an interesting admission from a well-known atheist, Richard Dawkins. The machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer engineering journal. It's surprising that somebody like that would comment on that, but I think it's an honest recognition. So as far as the third layer, as far as what is actually produced, remember that it's going to be proteins and protein machines. That's an example. And again, rather than try to explain, it's better to just have a video. Again, this is just like that last video. This is from Unlocking the Mystery of Life, which is by Lester Media, which is a great video to learn more about these.
The idea of irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice.
These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or a sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction. And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force, it has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's, it's not convenient that we give them these names. That's truly their function.
And something that was recently found was that they actually even include a clutch. So that's another part that's can be added to that list. So, wrapping up here, is Darwinian evolution, because it's a blind and unguided trial and error process, it is not efficient as a mechanism for generating the type of biological complexity that we see. I think that's pretty well laid out in some of the studies that I quoted. So there's just too much complex specified information in many of the proteins and the enzymes in biology to be generated by Darwinian processes on reasonable evolutionary timescales. So this is a significant problem. So what is the solution? Well, we need to look to a goal-directed process that can see into the future, look ahead, and find what solutions there are to achieve things like rare functional sequences. And basically we're talking about design here. And there's a great quote from Steve Meyer. Agents can arrange matter with distant goals in mind. In their use of language, they routinely find highly isolated and improbable functional sequences amid vast spaces of combinatorial possibilities. We know from experience, okay, so this is not through some sort of revelation or anything like that, not through some sort of doctrine. We know from experience that intelligent agents often conceive of plans prior to the material instantiation of the systems that conform to the plans. That is, the intelligent design of a blueprint often precedes the assembly of parts in accord with a blueprint or preconceived design plan. I'm a structural engineer, and I can tell you that that is an accurate description of the way that buildings are built. You have to come up with essentially what are often referred to as blueprints. We don't really use blueprints anymore, but the idea is that there's drawings that describe what the building should look like, and then that is what the contractor goes off of to build. Our experience-based knowledge of information flow confirms that systems with large amounts of specified complexity, especially codes and languages, invariably originate from an intelligent source, from a mind or personal agent. As we can see here, intelligence is really the only known goal-directed process which can generate high levels of complex specified information. So, based upon what we've seen, what is the mechanism that can best explain high levels of CSI? Would you say that it's intelligence or the mutation and selection mechanism? Well, we know that intelligence can produce high levels of CSI, whereas mutation and selection does not seem to have the ability to do so, at least with the probabilistic resources available. Therefore, we would say that intelligent design is the best explanation. We made it.